we have a couple of different ways that we can get stuff into the cell. Well, let's think, for example, what are some things that I want to bring into a cell? Why do I need to move stuff in? Okay, pretend that you are a cell. What sort of things do you take into you? Water, food, nutrients, oxygen, and then some stuff that I need to get rid of. Tell me some stuff that your body gets rid of. Do what? Toxins. How? We can sweat them out. What are some other ways that your body gets rid of stuff? I have stuff inside me right now that needs to come out, maybe.
Leaking.
Yeah, I'm gonna do my pee and my poop and my breathing. I breathe out. So when we're talking about this physiology, all we're referring to is how are my cells doing this? They don't have a little mouth. They don't have an anus. They don't have a urethra. They don't have pores. How do they get stuff in and out? This is what we're discussing. And this thing has got to go. I don't know what this is, but it is in my way. All right, so we have passive ways of getting stuff in. We got active ways of getting stuff in. Passive simply means we don't need to use any energy. We don't need any ATP. It's going to happen just because it's going to happen. I don't need anything in order to make it happen. I'm not having to shove it. I'm not having to push it. I'm not having to cram it. I just don't need any energy. And so, let me back up here. The 4 types of passive movement, meaning no energy needed: diffusion, facilitated diffusion, osmosis, and filtration. Talked about this a little bit on Friday. So I'm just going to go over it because I know I got a little bit of confusion. So we're just going to go back, kind of rework all of this. Some of the things that can impact the ability of stuff to get into my cells. Well, how big is the thing? Is the thing super duper big and I may need to be a little bit careful so I don't rip that cell membrane? Can it travel through fat? Well, remember, our cell membrane, our skin of the cell, is made of fat. It is a lipid bilayer. Some stuff travels very easily through fat, some stuff doesn't. What is the charge? What kind of ion is it? And finally, temperature. The hotter stuff is, the faster it's going to move. So those are the 4 factors for determining how easily and how quickly is stuff going to move. So the concept of a concentration gradient. Basically what we're referring to here is when we are moving stuff with a passive, no energy required, we are moving from a high concentration to a low concentration. The difference between these 2 concentrations is the gradient. I think we used the analogy on Friday that you are at a sporting event, you're at NRG, the Texans have triumphed, they are victorious, huzzah. Everybody's now leaving the stadium. We have a high concentration of people in the stadium, moving to the low concentration of the parking lot. I don't know about you, if anyone here has been to a Texans game or really any concert. At the end of the concert, it really just is you're shoulder to shoulder. You are very highly concentrated. And what? It's hot. It's smelly. People are intoxicated and they're smelling of VO and it's just sweat pouring off of you. I want to move pretty fast to the parking lot where it's nice and cool and I don't have this claustrophobia. I don't like people. Well, I don't like people touching me. I need my personal bubble. Just get away from me. And so the difference between the population in the NRG and the population outside, that's the gradient. Here we have another example, is a slide at the park. The taller the slide, the greater the difference between the tallest tall and the lowest low, the faster you're going to move. Here I have the lamest slide in the park, where I don't have a very big difference between my tallest tall and my lowest low. And this is one of those terrible, you know, the ones where you had to kind of shuffle yourself down the slide. Well, we all been to a park when you were a kid. I had a park like that, and it was made of metal. So you're shuffling yourself down and you're leaving like chunks of skin and look like a burn victim when you have finished. So this is the concentration gradient, just the difference between concentration high and the concentration low. So with diffusion, here's just a picture, we went through this. Great, another example is to have a fart, or the good smelly thing that you plug into your wall, and when you stand right in front of it, holy smokes, does it smell strong. But as it does what? Diffuses through the room, suddenly it does not smell nearly as strong. You have a much bigger area for those molecules to accumulate in. Some of our molecules are super duper big. We still want to move them in, but we don't want to damage the cell membrane. And so we have, sorry, invisible bug. We have facilitated diffusion. I'm going to move this thing down. We have facilitated diffusion. To facilitate simply means to help. So I might have these little doors, these little sliding doors. Here I've got, for example, a glucose, and that glucose needs to go in. That's what is keeping our cell alive. That's the nutrition that we're taking in. It's super big though, and I don't want to cram it through this membrane because it's going to break the membrane and kill the cell. And so I have this little door, this little protein that allows it to travel into the cell. But we're still not using any energy. We're still using the concentration gradient, and we have low concentration of glucose here, high concentration of glucose out here. We're moving it in, moving it in. That's what's providing nutrition for our cell. But we have a little helper. We have a facilitation. It's a great word, facilitate. Here's our picture of facilitate. So here's an old man. He's gone from the top of the stairs to the bottom. He's following the gradient, and we have a little helper to help him along. Osmosis. Same idea, except it only pertains to water. In this case, we're not moving the solutes. We're not moving the molecules. We're moving the water. So if I have an area of a super high concentration and a super low concentration, what the water is going to do is dilute so that both concentrations become equal. That's the whole idea of osmosis. We're going to move the water to dilute out the super concentrated, and so that both sides are equal concentration. Is everybody with me so far? Fabulous. Here's where I lost everybody. So I want you to erase everything from Friday. I lost everybody on Friday. We're going to talk about this today as if it's brand spanking new. We've got three terms: isotonic, hypotonic, hypertonic. All these words are referring to is what's the difference? Do I have an area inside the cell that is more concentrated than the outside of the cell? Do I have the area on the inside that is less concentrated than the outside? Are they equal? Iso means equal. So if I have an isotonic, it means both areas are equal. Nothing is being promoted to move. If I have hypotonic, the extracellular fluid is hypo. Tell me where the extracellular fluid is. Thank you. It's on the outside of the cell. If the outside of the cell is hypo, what kind of concentration am I looking at? does hypo mean? It's small or beneath. So what I have here, get my draw on. So here, hypotonic, that means the inside has got higher concentration than the outside. Does everybody see how that worked? So if the inside of my cell has a higher concentration and the outside of my cell has got a lower concentration, I'm going to be promoting the movement from where to where? Inside to outside, high concentration to low concentration. This is after the game. Everyone is inside NRG. We are moving outside to the parking lot. On the opposite effect, we have the extracellular, the outside of the cell is now hypertonic. Meaning what? The outside is higher concentration, the inside is lower concentration. How do we promote movement now? We're going to move into the cell. Let me give you an example of this that I think most of you will be able to relate to. This is specifically referring to a hypertonic solution. The outside is higher than the inside. All right, in your minds, some of you may not know what I'm talking about here, that's okay. In your minds, I need you all to go to the store and grab some gummy bears. Gummy bears, or worms, or whatever gummy you want, it doesn't matter. You're gonna put that gummy bear, this is a great experiment for those of you that are over the age of 21. You're gonna put those gummy bears into a Tupperware. Some of you are giggling because you know where I'm going with this. What do you think I can add to this Tupperware to make a delicious treat for later? Yeah, usually vodka. Vodka. Guys, I've just made, a hypertonic solution. So for those of you that are lost right now, this is how you make alcoholic gummy bears. Because when I add the vodka, I am now hypertonic, which means the outside of the gummy bear is more concentrated than the inside of the gummy bear. Tell me how I'm going to move. The alcohol is going to go into the gummy bear. What happens to the gummy bear? It gets bigger. Yeah, and if you accidentally forget them in there for like a week, holy smokes, I did that once and it was terrible. I had gummy bears just exploding. It was just a huge sticky mess. Still ate it though. Delicious. But that's a great example of hypertonic. It's also a great example for why do we care? There are IV fluids or intravenous fluids that will promote movement into a cell or will promote movement out of a cell, and that can help resolve some health concerns. So that's all we're referring to. Hyper, hypo, iso. What is the concentration on the outside compared to the inside? Does that clear everything up for everybody from where I kind of messed it up on Friday? That was all on me. Doug was just super tired. Everybody okay? How about you in TV land? Does that clear it up a little bit? I don't have my chat on, so if you have any questions or need me to clarify even more, let me know. Sweet. What is happening? All I want to do is move you guys. It won't let me move you. So I just do this. There we go. Let me move you. That's weird. Oh well, nothing I can do. All right, let me make sure. Hey guys in TV land, did that slide change? Are you looking at a slide that says the difference between the pressure of blood? Okay. So we have fluid in the tissue. We have fluid in the cells. We have fluid outside of the cells. Here we have a word, oncotic. Oncotic. This is simply the difference between the pressure of the blood and the pressure of the tissue. There are some times when I might accidentally promote excess fluid moving into the tissue. This is a medical term, you probably learned this already, called edema. Let me give you an example of when fluid moves out of the blood and into, now I'm not bleeding, okay? I'm not leaking blood, I'm leaking fluid. You ever gone on a super long road trip or been on a kind of a longer airplane ride? And you go and you take your shoes off, hopefully not on the airplane, because if you do that, you're a terrible, terrible person. But in the car, kick your shoes off, you stop for something to eat, you go to try and put your shoes back on. Sometimes what happened? Your feet swole up. You ever had that happen? No? Oh, it's because you're young. Well, no, because that used to happen to me when I was young. Do cross-country drives, and especially when you're flying. If you do a really long flight, sometimes your feet and your ankles will kind of swell up a little bit. That is because of this. It's promoting the flow of fluid out of the blood and into the tissues. All we need to know, though, is the term for it, which is oncotic pressure. All right, so the next type of passive movement, we're still doing passive movement, this is going to be filtration. I like to compare filtration to a coffee maker. Not the fancy Keurigs that you all use now. I'm talking about a good old-fashioned Mr. Coffee. You put the filter in, you put the coffee in, you fill the back of it up with water, you let it go. The water drips through the filter, carrying the liquid coffee with it, giving you a lovely cup. There are some things that can impact this. What happens, do you think, if I don't put enough water into the coffee maker? Is it going to filter properly? What about if I try and fill it up with a fireman's hose? Yeah, I'm going to probably bust it up. This is a great example of how our kidneys work via filtration. Our kidneys filter out our blood. They filter out toxins, put stuff back in that we need to reuse. Blood pressure affects the filtration rate of your kidneys. If your blood pressure is too low, it won't allow your kidneys to filter, just like our Mr. Coffee with no water. If your blood pressure is too high, you're going to start busting stuff out. Pressure has to be exactly right. The pressure that we're using in this coffee maker scenario is called hydrostatic pressure. Hydro meaning water. Yeah, good, water. Hydrostatic pressure. This is going to just simply push the liquid through, not needing any outside energy. All right, so those are the 4 passive methods of movement. Now we're going to discuss the active methods of movement, meaning that we do need to have energy. Why? Why is a passive route not going to work? Well, maybe it's not fat soluble. Maybe it's something that does not pass easily through fat, like, I don't know, water. Maybe the molecules are super, super humongous, and it's going to wreck the cell membrane if I just passively move it through. Or if they're on the wrong side of the gradient, that's probably the most common one. Okay, so let's go back to NRG Texans have won, huzzah. I am moving with the gradient from high to low, and I'm moving with the crowd, and I'm shoulder to shoulder, and I'm miserable. I'm just waiting to get out into the parking lot, and I realize, oh my God, I forgot t-shirt I just spent $180 on. So I have to turn around, and I have to try and go the opposite way. You see how that's going to expend a little bit of energy on my part? I'm going against the gradient. Now I'm going from low to high. That's going to take some energy. That's going to be active. So we're going to talk about some ways that we can use active energy to get stuff into a cell and get stuff out. Just like we just talked about. If we are cells, we need to take in oxygen and nutrients and water, and we need to get rid of excess toxins, undigested remains of your meal, excess water in your urine. So we're going to talk about how do cells eat, how do cells breathe, how do cells poo. Because cells do poop. We are just walking meat sacks of cell poo. You want to break down humankind to its lowest common denominator, that's exactly what we are. We're walking meat sacks of cell poo. You are welcome. All right, so active, remember it does require energy. If we have substances moving in the same direction, this is symport. Sim meaning together. Think of a symbiotic, working together. If we have substances moving in opposite direction, it's anti, meaning against or opposite. These active transport processes, we have stuff, endocytosis. Tell me, is this one going to be moving into the cell or outside of the cell? Endo. Going to be moving stuff into the cell. Exo. Yeah, that's moving stuff out of the cell. Endocytosis, I'm taking in food. I'm taking in drink. Exocytosis, I am peeing and pooing. Everybody with me so far? Fabulous. Oh, this is just another slide of describing, showing you a picture of me trying to go back and get my long gone t-shirt. Somebody picked that up an hour ago and took off with it. So you hang on to those or don't buy them at the game. Just saying. Not that I've never lost a Texans shirt. I had to mortgage my house to get one, so. Well, you guys are not receptive to my jokes at all today. My goodness gracious. I need to get you up, get you moving a little bit. All right, so active transport, as I said, moving against the gradient. That's all this slide is saying. Got a little ahead of myself in my lecture, got excited. All right, so let's talk about getting stuff into the cell, endocytosis. If my cell eats a solid particle, like me eating a Chipotle burrito, the term for this is phago, or phago, depending on who taught you. My instructor pronounced it phago. Other instructors here pronounce it phago. Both are correct. Phago means to eat. So if I have phagocytosis, let's break this down, medical terminology. We know phago is to eat. What is cyto? Cell, and what is osis? Condition or pertaining to. Phagocytosis is pertaining to a cell eating. We actually have a white blood cell. He's one of my favorite white blood cells because he's kind of schizophrenic. He's kind of like the Hulk. When he's just hanging out, his name is monocyte. And he's just this mild-mannered, you know, like top of the morning to, I don't know why he's English. This is got this little top hat and a monocle in my mind. But when a foreign invader comes in and we go Hulk smash, he turns into a big eater. In fact, his name becomes Macrophage, big eater. And he just goes around and finds foreign invaders and stuff and just goes, and he just eats it. And then he tries to get rid of it, or he'll sacrifice himself for you. I know, it's very dramatic. So when you're at home and you're like, I don't have any friends and nobody likes me and I'm feeling very sad and very blue, just know you have millions and millions of white blood cells who are willing to lay down their life for you. I know, it's endearing and very lovely. So this is phagocytosis, when we are engulfing solid materials. I love this picture. Little cell, and he's engulfing all of these little solid, I don't know what those are, little viruses, little bacteria. Pinocytosis is taking in liquids. The way that I remember this is there is a type of wine. Tell me what kind of wine I'm thinking of. Do what?
Pinot Grigio?
Not Pinot Grigio. So when I think of pinocytosis, I envision myself, although I'm not a wine drinker, but I envision myself with a wine glass, sipping on that Pinot, having a nice Pinot. Pinocytosis is when we are taking in liquid. So phagio is solid, Pinot is liquid. Finally, we have our receptor mediated. What this means is that unless you have the key, you are not getting in. So put in your mind now, my car is a cell. And I am something, we are something that wants to get into that cell. We need to get in. Can any of you guys get into my cell car? Hopefully not. Yeah, the correct answer is no. The correct answer is no. Because what do you not have? Do you have what?
A brick.
Really? Mikhail? Thank you. You don't got one of these. My key. This is the receptor. I can put this into the lock of my door and I can open it, and that allows me to enter my cell car. I can't go to one of your cell cars and open it up with this key. I can't enter the cell, so I have to have the key to open the lock. That's the idea behind receptor mediated. Now, could I open the cell car with a brick? Yes. But what does that do to the cell car? It damages or destroys it. And then you're in big trouble because I will hunt you down because I love my car. I love my car. So that's receptor mediated, lock and key. So here are just 3 pictures showing everything I've just talked to you. Here I have phagio, meaning solid, I'm eating it. Here I have a big blob of something, this is Chipotle burrito that my cell wants to get into his belly. We go ahead, we come in contact with that cell membrane. We then rejoin the cell membrane and bring the food into the cell. Here is my liquid denoted by these little blue squares or diamonds, depending on what side they're turned on. Exact same thing, but with liquid. We bring it down, we come in contact, we then chop off and bring it in that little vesicle. Here I need to have my key. The star is my key. I need to insert that into the lock. That's what then allows me to come into the cell. What goes in must come out. So here I might have toxins, I might have waste, but another thing I might have are product. I'm making proteins, I'm making stuff, I'm shipping it out with my Golgi apparatus. I might have things that cell created that I have to get out of the cell and ship it to other parts of my body. This is going to be exocytosis, or outside of the cell. This is the exact same process, but in reverse. Remember when we talked about the Golgi apparatus and how it puts the FedEx shipping label on it? It then sends it in a transport vesicle. It then binds with the lipid bilayer and then releases its contents outside of the cell. And yes, I did Google image search a pooing dog for your viewing entertainment. Honestly, if I don't even know how many watch lists I'm probably on right now because of my Google image searches. I know I have an FBI file. I'm not sure what other government agencies are keeping track of me. I do feel kind of bad for the guy that's got to look through all my computer searches, because out of context, pretty messed up. You know, I Google image searched the genitalia of a hookworm. Not like just for random fun, guys. I had a student who wanted to see what the genitalia of a hookworm looked like, so I went ahead and I did that. I took that bullet for you. Anyone else in here have an FBI file? Just me? Really? Are you all wondering why I have an FBI file now? If I tell you, I have to kill you all. Kind of the reason why I haven't. No, I'm kidding. I'm totally kidding. I'm totally kidding. I am not a murderer that you know of. No, I'm an immigrant. Every immigrant has an FBI file. I'm here. I have a green card. I'm not a U.S. citizen. So if you have gone through the immigration process at all, you got to file with the FBI. So it's not as provocative as you were thinking. All right, before we get into this next little bit, I do have a shout out to Mikhail, a brick. All right, I have a little video that just kind of reiterates everything I've just talked about. It is kind of an elementary video. It's this little animation called the Amoeba Sisters, and it's adorable, but it does kind of go over everything we've just talked about, give you a nice little overview before we get into not to show you the test that is on my computer right now. All right. Oops, that's not what I want. All right, so this is just an adorable little video. All right, can you guys in TV land see that? It says cell transport. All right. Did you hear that? Okay.
Have you ever wondered what it might be like to be inside a cell? Imagine the genetic material, the cytoplasm, the ribosomes. You'll find those in almost all cells, prokaryotes and eukaryotes. Eukaryote cells, in addition, have membrane-bound organelles. All of those structures and organelles have different functions, but cells are not isolated little worlds. They do have a lot going on inside them, but they also need to interact with their environment. It makes sense that to keep a stable environment inside themselves, otherwise known as keeping homeostasis, they must have some control on what goes in and what goes out of them. A very important structure for this that all cells contain is the cell membrane. By controlling what goes in and out, the cell membrane helps regulate homeostasis. It's the homeostasis king. Let's take a look at the cell membrane. You could have a whole course on the cell membrane itself. It has amazing structure. It has signaling abilities. But to stick to the very basics, it's made of a phospholipid bilayer. Bilayer means two layers, so you have these two layers of lipids. Now, these lipids, they're called phospholipids, well, they have a head that is polar, and they have a tail that is nonpolar, making them quite unique. Some molecules, they have no problem going through the cell membrane, and they directly go through the phospholipid bilayer. Very small, nonpolar molecules, they fit in this category. They're a great example. Like some gases, oxygen and carbon dioxide gas, those are great examples. This is known as simple diffusion. Also, it doesn't take any energy to force those molecules in or out, so it's known as passive transport. Simple diffusion moves with the flow, meaning it moves with the concentration gradient. Molecules move from high concentration to low concentration. So when you hear someone saying that something's going with the concentration gradient, that's what they mean. They mean it's going from a high concentration of molecules to a low concentration of molecules. Now, remember how we said the cell membrane is actually a pretty complex structure? Well, one thing we haven't mentioned yet are proteins in the membrane, and some of them are transport proteins. Some transport proteins act as channels. Some of these proteins actually change their shape to get things across. Some of them open and closed based on some kind of stimulus. All of these are good things because it's helping with molecules that may be too big to cross the membrane on their own, or molecules that are polar and therefore need the help of a transport protein. This is known as facilitated diffusion. It's still diffusion, and it still moves with the concentration gradient of high to low. It doesn't require energy, though, so it's also a type of passive transport. It's just the proteins are facilitating or helping things pass. Charged ions often require a protein channel in order to pass through. Glucose needs the help of a transport protein to pass through. In osmosis, for water to travel at a fast rate across the membrane, it passes through protein channels called aquaporins. So these are all examples of facilitated diffusion, which is a type of passive transport and moves with the concentration gradient of high to low concentration. Now all the transport we've mentioned has been passive in nature. That means it's going from high concentration to low concentration. But what if you want to go the other way? For example, the cells lining your gut, they need to take in glucose. But what if the concentration of glucose in the cell is higher than the amount of glucose concentration in the environment? We would still need to get the glucose in, so it's going to have to be forced against the regular gradient flow. Movement of molecules from low to high concentration will take energy because that's against the flow. Typically, it's going to require ATP energy. A reminder that ATP, adenosine triphosphate, it has 3 phosphates. And when the bond for the last phosphate is broken, it releases a great amount of energy. Yeah, so ATP is a pretty awesome little molecule. ATP can power active transport to force molecules to go against their concentration gradient. And one way it can do that is actually energizing the transport protein itself. One of our favorite examples of active transport is the sodium-potassium pump. So that's definitely something worth checking out. There's other times a cell needs to exert energy for transport. We're still in active transport right now, but let's say a cell needs a very large molecule. Let's say a big polysaccharide. If you check out our biomolecule video, that's a large carbohydrate. Well, you may need the cell to fuse with the molecules it's taking in order to bring it inside. And this is called endocytosis. Think endo for in. Often this fusing of substances within the cell membrane will form vesicles that can be taken inside the cell. Endocytosis is kind of a general term. There are different types of endocytosis depending on how that cell is bringing substances inside. Amoebas, for example, rely on a form of endocytosis. In this example, pseudopods stretch out around what they are going to engulf, and then it pulls it into a vacuole. And there are other forms too, such as the fancy receptor-mediated endocytosis. This is where cells can be very, very, very picky on what's coming in because the incoming substances actually have to bind to receptors to even get in. Or pinocytosis, this allows cells to take in fluids. So to the Google to find out more details of the different types of endocytosis. Exocytosis is the reverse direction of endocytosis. Think exo and exit. They sound very similar too. Exocytosis can be used to get rid of cell waste, but it's also really important for getting valuable materials out that the cell has made. Want a cool example? Well, think back to those polysaccharides. Did you know that large carbohydrates are also really important for making plant cell walls? Cell walls are different from cell membranes. I mean, all cells have membranes, but not all cells have a wall. But plant cells do. And if you're going to make a cell wall, you're going to need to get those carbohydrates that are produced in the plant cell out of the cell to make the wall. So there's a great example of when you'd need exocytosis right there. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So again, pretty simple, pretty simplified, but it's definitely a nice little animation. Yeah, it was not skipping. It's a stop animation. More photos than anything. All right, do I have any comments, questions, concerns about anything transport related? Did any of you guys bring your dogs today? Are you going to? Probably not. I haven't decided if I'm gonna bring Poppy in. She was so sad, guys. Oh my God, it broke my heart. Because Maisie is her BFF. And although Maisie kind of is rethinking all of her life decisions when this happens, but yeah, Maisie and Poppy are, well, Poppy thinks Maisie is her BFF. So usually when I go put the leash on, we're going to my significant other's house, and Poppy absolutely adores him. Thinks he's just the bee's knees. She gets super excited. She starts spinning very quickly. Just spin, 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 and then she goes and runs in her bed because she knows that I have to go now pick her up. She's a hot mess. So she sees me get the leash, and she sees me put it on Maze, and she gets super excited, and she goes and she dives bombs in her bed, and she rolls over, and I'm like, baby girl, you're not coming with me. So I take Maisie, and all I see is Poppy with her tail tucked and her ears back, just standing in the kitchen watching us go. It was the most heartbreaking thing I've ever seen. So I may have to bring her one day and just let her hang out for a while. You guys can help me socialize her a little bit. That might be fun. All right. Now that we've moved stuff in and out of our cells, now we're going to talk about what happens when cells need to replicate. We have two different ways that I can create new cells. This video back up. I have two different ways that I can create new cells. I have meiosis and I have mitosis. Meiosis is where I take part of one cell and part of another cell, I mosh them together and make a new cell. Meiosis is you. You are a product of meiosis. You had some of your dad's DNA and some of your mom's DNA, and bippity boppity boop, you are created, but you are a unique cell. Your mom is a product of her mom and dad's DNA, et cetera, et cetera. So right now, you are a genetic stew from every contribution to your lineage through the end of time. This is why sometimes you might have a child and you look at the baby picture and you're like, oh my God, look at this. Here's a picture of our great-great-great-great-grandmother and their carbon images. Because of this stew, this unique DNA, we're not going to be concentrating on meiosis. You'll get that in another class. We're going to be concentrating on mitosis. And what mitosis is an exact replica. Why is this important? Well, let's talk about our livers for a minute, because it's the first organ that came to my mind. Your liver, those cells don't live forever. And sometimes you might do a little bird damage to your liver, eat too many of those gummy bears, and the cells need to replicate. But I want the new cell to have the blueprint and have the work manual from the old cell. I want a liver cell to work like a liver cell all of the time. Because when I start getting cells that don't know how to work like a liver cell, bam, I've got cancer. That's basically what cancer cells are. They are cells that are replicating that don't know how to do the job in the area that they're being replicated to. And so now I have all of these liver cells who don't know how to function like liver cells, and my liver doesn't work anymore. So it's super important that I have an exact copy from one cell to another. And this is going to be mitosis, an exact replication. So we have a bunch of different phases of mitosis. The first phase is called interphase. And this is the phase that the cell spends the majority of his life. We are not replicating. We are not even getting ready to replicate. We are growing, we are maturing, and we're just doing, if we keep with our liver, this is just the liver cell doing liver cell stuff. He's just walking around, doing liver stuff. He's happy. He's a contributing member of his community. He is just in cell kind of stasis. He's just doing his own thing. He's doing his own job. So this is the interphase where the cells spend the majority of their life. Cells don't always divide. Cells have to be told by your body to divide. And that's only when we need more cells. It's also why I don't have like a body-sized liver, because my cells are not just uncontrollably reproducing. At some point, I have enough liver cells, I don't need them to reproduce anymore. So interphase is just where we're just going about doing our little cell jobs, spending our little cell day. So interphase, this is the period between divisions. Again, we're not actively dividing, we're not even getting ready. Now, originally, we called this the resting phase. We didn't originally call this the interphase. We called this the resting phase until we realized that cells are never truly at rest, right? All this phase means is we're not dividing. It doesn't mean that he's not doing anything. He's not accomplishing his life goals. This cell is still, having purpose and having a job. So resting phase was kind of a not correct term. So what we did was we changed it to metabolic. Metabolic just means we're going about our own cell business. We're just simply not dividing. If anything else you want to know about the definition of interphase, definition is we are not dividing. We have our nucleus, our DNA is contained within that nucleus. All of our organelles are functioning just as they should. Nothing else is happening. Now the magic happens. My body has signaled, oh my gosh, she did some liver damage this weekend. Gave myself a little bit of hepatitis on Saturday and Friday. And so I got some liver cells that I got to replace. Now I'm going to go in and I'm going to start replication. The really important thing with this mitosis, though, is through this process, I need the DNA to be exact copy, an exact replica, so that each of my cells, the old cell and the new cell, have the exact same working manual. And the new cell knows how to do the job just as the old cell did. So this DNA has to be replicated first before we can start dividing. Time flies when we are having fun. I'm going to go ahead and end it here before we get into all of this mess. Oh yeah, this is where the magic happens.